Hello, and welcome to our recorded lecture for Population Genetics. This is section 2 of chapter 23, and we're only going to be covering section 2 of chapter 23 here. The rest of chapter 23 is going to be covered towards the end of the semester when we talk about evolution. Now, in population genetics, we are going to be clearly talking about the genetics of populations. And you might be wondering, well, why would this matter? Many of you are interested in one way or another in medicine. And in medicine, you may study genetic diseases, or you may deal with people who have genetic diseases, or the results of certain genetic diseases. And we know that some genetic diseases are more common in some certain populations. A good example of this would be how sickle cell anemia is more common in people of African American heritage or African heritage. In the United States, African Americans tend to be the group that has it, or people who are somehow having African ancestry. Do other people have it? Potentially, but it's going to generally follow a certain inheritance pattern. You might wonder, well, why would we want to study these things? How would we want to know whether or not it happens? And why do we care about these? And it may be that we discover that a lot of genetic traits tend to be seen in a small portion of the population. Some become more common, some become less common. And studying population genetics allows us to understand how traits change in a population. It allows us to understand what is common and what is recessive. And for those of you who have more of an environmental interest in biology or an animal interest, we also use population genetics to look at how we might want to go about breeding certain animals in captivity together in order to increase or maintain the genetic diversity and to figure out which ones we probably should not mate together. For sickle cell, we've discovered that why it is common has a lot to do with a concept known as heterozygote superiority. If you have the normal red blood cell shape, then your red blood cells carry a lot of oxygen. This is good. But if you get malaria and you have normal red blood cells, you're probably, without medical intervention, not going to make it. If you have full-blown sickle cell anemia, your cells, instead of being round, your red blood cells, they are crescent moon or sickle shaped. They're not going to carry oxygen as well. It's going to lead to a shortened lifespan, but it's also going to lead to a whole host of other medical problems. However, you won't die of malaria. Now, if you're a heterozygote, so you have both traits, you're really lucky. You have enough normal red blood cells to not be affected by sickle cell anemia, but you have enough sickle-shaped cells to not die of malaria. It's a better trait to have then in areas where malaria is common. And I wonder where malaria is common. It's common in Africa. So it would make sense then that these cells, or that the traits for sickle cell anemia are going to be more common in people who came from Africa. In population genetics, we look at changes in genes in a population. We look at how genetic percentages change in large populations over time, how common certain traits are, and a variety of other factors. Before we talk about population genetics more, we should actually talk about what a population is. So a population is a group of individuals of the same species that live in the same area, or they occupy the same environment. So we could right now talk about the population of Eastern University students versus the population of Cabrini students. Or we could talk about the population of dogs in the Philadelphia area. We could talk about white-tailed deer populations on the East Coast. Generally speaking, with a population, it's going to be a group of the same species, and you'll have to understand something about their location, or what are the boundaries of where these people, or animals, or plants, bacteria, etc. are. So then population genetics is the study of genes and genotypes in a population. It is going to study genetic variation within the gene pool. So it's going to look at how that genetic variation is, or what that genetic variation is, why it exists, how it's maintained, how it changes over time, how it changes from generation to generation, how it leads to phenotypical variation, because as you've learned so far in genetics, we can talk about genotypes, so genetically what you are, or your phenotype, what you physically look like. The concept of population genetics emerged in the 1920s and 1930s from a mathematical standpoint by mathematicians and biologists looking at the work of Mendel, Darwin, and a few other people and bringing it together. You know that each individual has two alleles for any given trait, at least in something where they have two sets of traits. And because we know this, it means that we can look at the variation in alleles in a population even when we don't even see that level of variation, because, well, there's two traits for everybody. So sometimes a dominant trait and a recessive trait are going to be in a heterozygote. And you may not be able to see it, but population genetics is going to tell us how many heterozygotes you can expect given a certain number of people with dominant and recessive traits. It's pretty cool the way this works. And it will allow us to look at variation in alleles and populations. To do this, we also need to consider the concept of a gene pool. Because a gene pool is, yes, the pool of genes or pool of alleles, but it's important to look at from a reproductive structure as well as from a conservation management structure. If you reproduce, your genes are included in the gene pool of the next generation. If you don't reproduce, your genes are not going to be included in the gene pool of the next generation. So an animal that dies off, if it didn't reproduce, its genes are not included. If an animal never finds a mate, its genes are not included. But those that do reproduce, their genes are included in the gene pool for the next generation. Populations themselves are dynamic. They change. They're not static. They're not going to stay the same forever. There's going to be constant changing in a population. Populations do exist over a large area. Sometimes they subdivide into smaller populations. And sometimes those populations become really distinct from each other. Maybe there's a geographic barrier. A river, an ocean, mountains, some type of a barrier. For some things, it could be a road. A large highway is a good barrier for a small insect. And geographic barriers are going to lead to isolation of populations. As populations become isolated, they interbreed, and we find that the populations become more genetically separated from each other because each population adapts to better live in its own environment. They become better suited for their environment because if you're better suited toward your environment, you're more likely to live longer, to live long enough to reproduce, and then your offspring are more likely to reproduce as well. So those genes become more and more popular in the gene pool, or more and more prevalent really in the gene pool. And the ones that are not prevalent, that are not helpful at surviving in that area, become less prevalent. What might be some examples of things that are going to change in a population from generation to generation? Well, the size of a population might change. The location of where an animal is might change. Maybe the ratio of males to females could change too. Sometimes we find that changes are going to result in genetic shifts and adaptation because the genetic composition is going to change as things interbreed with each generation. So if a population ends up in an area that is suddenly colder, let's say we're talking about a mammal here, that ends up in an area that is suddenly colder than where they lived before, those that are genetically predisposed to grow more fur or more hair, they're more likely to survive than those that grow less. So you would see that our gene pool would then start to favor those with more hair. If they go from an area with lots of food to a scarcity of food, a lot of your animals may not survive. So those that are better hunters, that have better eyesight, they'll survive. We're talking about prey, and suddenly there's a whole lot of predators nearby. Those that hide better, the prey that are faster, they're going to be the ones that are going to survive. So certain traits allow certain organisms to have a better competitive advantage to not only survive, but then to reproduce. And you might wonder, well, how do genetics play in reproduction here? I'm going to use a couple bird examples because if you did not know this, I teach our ornithology class. And in there, we talk a lot about bird behaviors, yes, but fortunately or unfortunately for me, a lot also about bird breeding behaviors. I'm going to use a couple that I think you've, well, one I know you've seen, some you might not have. So peacocks. You've all, I imagine, seen a peacock before. They have these brilliant, the males, these brilliant tails, beautiful tail feathers. The more attractive the male peacocks are, the better they are at strutting around and flaunting those tail feathers, the more attractive the females are going to find them and the more likely they are to mate with them. So a male who is genetically, has a higher level genetically of feather health and brilliance in color is going to get more mating opportunities than a male who doesn't. And a lot of other birds, it's about song. They're better singers. They have the ability to learn more songs. They're going to become more successful at reproducing. We find that genes in populations are usually polymorphic. A polymorphism is the idea that many traits display variation within a population. So your polymorphisms are when there's more than one trait, more than one morph, if you will, of what the population looks like. These are due to two or more alleles influencing a phenotype, meaning that there's one or more phenotypes possible. So in the pea plants that you did with Mendel, there was short and tall. There were purple flowers or white flowers, yellow seed pods or green seed pods, and so forth. A polymorphic gene occurs when there are at least two alleles, both of which have a frequency of greater than 1% in the population. We find that a monomorphic gene is one that exists predominantly as a single allele, meaning that more than 99% of the alleles in the whole population are that. So in a monomorphic, you're not going to see both traits displayed. In a polymorphic situation, you are going to see both displayed. So what causes polymorphic genes? Some of these are things you already talked about, the concepts of deletion, duplication. So as they're going through meiosis and the genes are being copied and things, sometimes pieces get lost, sometimes extra pieces get added. But we also can have polymorphisms due to the change of a single nucleotide. And we're going to see a little bit more about that before we get to our math. And the picture you see at the bottom of this screen are orchids. Same species, different colors. Polymorphism. So single nucleotide polymorphisms is your smallest possible type of genetic change in a gene. It is one single nucleotide different. An A instead of a T, and so forth. A one-letter change. Keep in mind that genes are a lot of base pairs. Genes can be 2,000 to 3,000 base pairs. And we find that in most genes, there's an average of 10 single nucleotide polymorphisms, or SNPs. SNPs that occur in more than of 1% or more are found frequently. So if there's something that is more frequent, you're going to find it more often. But 90% of the variation in the human gene sequence is due to these SNPs. So there's some gene variation due to larger changes. But a lot of what happens is due to a single nucleotide change. Things like sickle cell anemia are due to a single nucleotide difference. Many SNPs relate directly to human health. They may cause genetic diseases. It's also going to change how your body responds to different types of medical treatments, different types of medications, and it's one of the reasons why we're looking into the concept of personalized medicine, something that is best for your body and your genetic makeup. SNPs sometimes can tell you whether or not you're likely to have a genetic disease or whether or not something could be triggered in you in the future. And if so, we can start doing preventative measures if we know that there's going to be a certain problem. But these SNPs are important. They allow us to not only have genetic variation, but they can also allow us to track diseases, and they can help us sometimes narrow in on an individual to find out more about somebody based on matching their DNA to themselves or to a large database to find out how they're related to people. And now we're moving on to math. I know some of you are groaning at home. Sorry about that. But math is important in science. And we're going to do some relatively easy math as we go through this. The first thing we're going to do is look at the frequencies of alleles in a quantitative way. We can do this by calculating an allele frequency, which is the number of copies of a specific allele in your population, divided by the total number of alleles of that gene in the population. But we can also look at a genotype frequency, which is the number of individuals with a particular genotype, divided by the total number of individuals in the population. These are related equations, but they're going to give very different answers. Because remember, everybody has two alleles, but we only have one genotype. So how do we calculate these things? Well, you're going to see a lot of letters P and Q today. Because if there are two alleles at a locus, so at a gene locus, the location for a gene on the chromosomes, we use P and Q for representing the frequencies. The frequency of all alleles in a population has to add up to 1. Well, 100%, which is 1 expressed as a singular number. So if you only have 2 possible alleles, if you add up all of one and all of the other, you're going to get to 1. So P plus Q equals 1. Got that? P plus Q equals 1. If we're just talking about the frequency of alleles, this is our equation. We are going to look at genotype frequency versus allele frequency a little bit here. In this, you can see that we have a group of cheetahs. Quickly, let's see how many cheetahs you can find. I know a bunch of you are going to tell me that you only found 4, but if you look, there are 6. You should hopefully be able to find them by looking at where the arrows are pointing. And in this, let's count. How many large A's are there? Hopefully you see that there are 7 capital A's. How many lowercase a's are there? Hopefully you're seeing that there are 5 lowercase a's. Now we knew that there were 6 individuals, so we knew that there were 12 possible alleles. To calculate P, we would say 7 divided by 12, because P is going to stand for our dominant trait, so our capital letter. So 7 capital A's means P is 7 divided by 12, which is 0.58. Q is standing in for our recessive, and that would be 5. 5 divided by 12 is 0.42. When we add up 0.58 and 0.42, look at that. We get 1. P plus Q equals 1. You will find that sometimes if Q is very, very small, we might estimate that P is equal to 1, but we're not there in this example. And if you want to follow through how we calculate the frequency of our genotypes, that's what we're going to do next. Now, an allele in the P plus Q equals 1, an allele is just one letter. A genotype is two letters. It's either capital A, little a, or a capital A and then a lowercase a, or little a. So we're going to see that this is going to get a little bit more complicated with our equation here. To get the frequency of the cheetahs with the homozygous dominant capital A genotype, we would take the frequency of P, which was 0.58 times itself. And that gives us 0.33. If you want to follow along with your calculator, you can do the math. So we can then calculate the frequency of a homozygous trait, or of the homozygous genotype, homozygous recessive genotype. That 0.42 times 0.42 is 0.18. Now, hopefully you're thinking, wait a minute now, we still need to calculate one other one. What about the heterozygote? And that's true. And we calculate our heterozygote, which can be either big A, or little a, big A. So we calculate that by multiplying the 0.58 times the 0.42, and then adding to that the product of 0.42 times 0.58. When we add those together, we get 0.49. 0.33 plus 0.18 plus 0.49. What do you think this is going to add up to? Hopefully, you're saying it probably is going to add up close to 1. We are going to come back to the math in a moment, but first we're going to talk about Hardy-Weinberg equation, the equation that we just about got to there. This is a mathematical equation that is used to calculate the genetic variation of a population at equilibrium. In 1908, G.H. Hardy and Wilhelm Weinberg independently, completely separately, described the basic principle of population genetics, which is now named after both of them, the Hardy-Weinberg equation. One is an English mathematician, the other was a German physician. They came up with this concept at almost the same exact time. It's going to describe the relationship between allele and phenotype frequencies at equilibrium. In equilibrium, your population is not evolving. No one is entering, no one is leaving. The population is just constantly interbreeding inside of itself. Their equilibrium is the amount of genetic variation, or the concept of their equilibrium is that the amount of genetic variation in a population is going to be constant from one generation to the next, as long as there are no disturbing factors like evolution, immigration, emigration, mutation. So if nothing's going on, if everybody is just constantly breeding inside of that group, you're at the Hardy-Weinberg equilibrium state. This equation is going to predict allele and genotype frequencies should remain the same from generation to generation if your population is at this equilibrium status. Now there are 5 traits for a population to be in the Hardy-Weinberg equilibrium. The first one, no new mutations. The second one, no natural selection. The third, the population is so large that allele frequencies do not change due to random sampling error. We'll come back to that in a moment. Point 4, no migration. So that means no immigration, no emigration. No one leaves, no one comes in. And then 5 is random mating, the idea that any one in the group can mate with any one in the group of the opposite sex. That random sampling error, it is important that we mention a little bit about that, because if that's not a concept you're familiar with, it really doesn't make any sense. Imagine, if you will, that I had a bowl of M&Ms. And in this bowl of M&Ms, there were 100 M&Ms. 95 of them are red. 95 out of 100 are red. And then there's one yellow, one orange, one blue, one green, and one brown. So you have 95 red, one yellow, brown, orange, green, blue. I tell you to reach in and grab 10. You grab 8 red, 1 green, and 1 yellow. And I tell you, I want you to tell me what you think the rest of the 100 are like. Well, you're going to tell me, okay, well, 80% of them are red, 10% are green, and 10% are yellow, and that there's none of the other colors in the bowl because you did a random sampling. And your random sampling has led to you to an error about what really exists. And a random sampling error, it's the idea that your population could, by choosing a couple of them, completely change your mind of what the actual population looks like. So a population does need to be large enough so that allele frequencies do not change due to a random sampling error. And each of the alleles needs to be large enough for the random sampling errors to not happen. Let's think about this for a moment. A population with no mutation, no natural selection, no migration, random mating only, and a really large population. Do you think we find populations like that often? We don't. We don't find populations like this. The Hardy-Weinberg equilibrium does not exist. However, we still utilize the equation to let us know when things are completely out of equilibrium. And we can utilize the equation to calculate allele and genotype frequencies. So how does this equation work? Well, for any 2 allele locus genes, P is your dominant allele, Q is your frequency of the other allele. And notice the same equation we just had before, P plus Q equals 1. And then we get this other equation. So P plus Q equals 1 is our frequency of alleles. What about our frequency of genotypes? Well, that is P squared plus 2PQ plus Q squared equals 1. I once had a student, actually, I think I've had a student ask this every year. Are you ever going to ask me to solve for what Q is without knowing what P is? No. You're always going to know either Q or P or PQ or something. So you're never going to have to solve this without any numbers. You will always get at least some of the numbers. So hopefully if you were worried, that's going to make a little bit more sense for you. We're going to help you get the numbers. You'll either be given the numbers or you'll have to count them from a population. But it will work. And now we're going to use these equations. So I'm going to go through some examples with you here, and then we're going to go through some examples in class. Because this is a topic where if you get it, you're fine. If you don't get it, it's easy to really be confused. And before we do, just a couple more things about your equations, just so you can use these as reference slides to come back to. So that P squared is your population that is homozygous for the first allele, usually the dominant one. 2PQ, that one is your heterozygote. It has both the P gene and the Q gene. And then your Q squared is your part of the population that's homozygous for your second allele, so that recessive allele. P squared plus 2PQ plus Q squared equals 1. Remember, P squared plus 2PQ plus Q squared equals 1. This equation is looking at flower color. In this, the CR is standing for your red, the CW stands for your white, and in this flower example, the CRCW is actually your pink flowers. So your red flowers plus your pink flowers plus your white flowers equal 1. That's 100% of your population. Those are the only possible colors that there are, so that has to equal 1. Let's look more at this. These plants that we're using are the 4 o'clock plants, where red and white are separate colors. And when they interbreed, you don't get red or white plants necessarily. You also get pink. Pink is your heterozygote. In pink, it produces both red and white color. And as you learned in elementary school, red plus white equals pink. So this is an exception to our Mendelian understanding of genetics. And let's say we had the flowers and your job was to go out and count them. And you counted 49 red flowers and 42 pink and 9 white. But let's say you then wanted to find out, well, what's the frequency, allele frequency of CW? You could calculate it. It's going to eventually be 30%. And you might be wondering, well, wait a minute, I don't understand, how is that possible? Well, you know that both of our white plants, both alleles are going to be white. CW, CW. So what you're seeing at the top is your frequency of CW equals. And then the top line of that shows CRCW. Every pink flower contains 1 white trait, 1 white allele. So we're doing 1 of that. So the pink plus 2 times the whites, because the white only have white alleles. So we get 1 from the pink, and then we get 2 from each white flower. So that's 42 plus 2 times 9. And then we're going to divide that by the total size of your population. Well, for this, your total number of alleles. So this is going to give us 60 for the top divided by 200. Because remember, there's 200 alleles in a population of 100 because everybody has 2 alleles. And 60 divided by 200 is 0.3 or 30%. So 30% is your allele frequency of CW. 30% of your population has a white trait, a white allele. The gene frequency here is a lot easier than the allele frequency. 9 out of 100, 9%. We're going to do some more examples. You really need to think through this and work on this. This is not, for some of you, going to be the easiest topic that we do. Some math, yes, but you need to understand the math we're doing. So now we're going to calculate the allele frequency of CR. And we're going to calculate the genotype frequencies of CRCR and CRCW. If you remember, the allele frequency calculation is P plus Q equals 1. Hopefully you remember on the last slide that we got the allele frequency for CW. It was 0.3. So what do you think the allele frequency for CR is? Hopefully, you're going to think that I'm going to take 1 and I'm going to subtract 0.3, and that's going to give me 0.7. And the answer is 0.7. I would draw it on here, but I'm kind of afraid it won't work. You might be wondering, okay, what if I want to calculate this a different way? Well, then you would say 49 times 2 plus 42 divided by 200, which is still going to give you 0.7. Let's calculate the genotype frequency of CRCR. Well, we know there's 49 of those plants, and we know there's 100 plants total, so it's 49%. What's the genotype frequency of CRCW? There's 42 of them out of 100, 42%. We're going to keep coming back to these. We're going to go through a lot of examples. Back to this slide about the Hardy-Weinberg equation. P squared plus 2pq plus q squared equals 1, where our p squared is our genotype frequency CRCR. Our 2pq is the genotype frequency for CRCW, and q squared is your CWCW. We know that p equals 0.7 and q equals 0.3. Even if that's all we had, even if we only had that allele frequency, we can calculate the genotype frequency. If p is 0.7, then the genotype frequency has to be 0.9 because it's 0.7 squared, or 0.7 times 0.7. And for 2pq, for the pink flowers, that's CRCW, you would say 2 times 0.7 times 0.3, which is 0.42. And then for the white flowers, that's CWCW, it's q squared, which is 0.3 squared, or 0.3 times 0.3, which is 0.09. Now, I know some of you are not good at math, so I'm going a lot faster than you can with your calculator. That's one of the reasons I gave you this as a recorded lecture. So you could pause it, you could stop it, you could do the math, and figure out whether or not you're doing things right. Take advantage of this as you go through this. Give yourself the opportunity to work through the math. I'm going to intentionally kind of pause speaking sometimes for a moment so that you get the opportunity to pause without missing anything and work through the math on your own. Now we're still working with our CRCR, CRCW, CWCW, our 4 o'clock plants, our red, pink, and white plants. And we still have the same information, the 0.7, the 0.3, and we have our frequencies of their generations. The 0.49, 0.42, and 0.09. So this is another way of looking at our flower. Now we're going to try something totally different. I'm telling you that green is the dominant trait. Green is big G. Little g is purple. I want you to determine the frequencies of big G and little g in this population. So, you might be wondering how do I do that? And I'm going to walk you through this step by step, giving you time to pause it. I want you to count how many squares total there are. Hopefully your number is 36. There are 36 total squares. Now I want you to count how many purple squares there are. Hopefully you counted 9 purple squares. So we know that 9 squares have a little g association. That means that q squared equals 9 divided by 36. Now if you say q squared is 9 divided by 36, that's going to tell you that q squared equals 0.25. Hopefully some of you are really good at math. If you know that q squared is 0.25, you also know you can take the square root of q, and take the square root of that number, and figure out what q is. So q is 0.5. So if we know that Q is 0.5, what's P? Hopefully you're remembering. P plus Q equals 1. So if Q is 0.5, what's P? Well, it's also 0.5 because 1 minus 0.5 is 0.5. So then we can take that information and we can calculate what percentage of these squares would be big G, big G. Well, if we know that P is 0.5, then P squared is 0.25. How about 2PQ? Well, P is 0.5, and Q is 0.5, and then 2 times 0.5 times 0.5, well, that's 0.5. So 50% of our squares are heterozygotes. Work through this. Figure it out. Make the math make sense to you. This is another one for you to work through. This is the same thing that we just did. If you need to practice it again, I'm going to leave this one in here so that you have the opportunity to practice through it without thinking that you could practice this and then go back and hear the last slide for the explanation. And here we have a totally different sort of creature to look at. We're moving from boxes to dragons, and you can thank Dr. Loxo for this example. She found it for us. Winged is our dominant dragon trait. So we're going to calculate the frequencies of winged versus non-winged, and we're just going to look at our allele frequencies here. So how many dragons total are there? Hopefully you counted 30. How many dragons do not have wings? Nine. So we have 30 dragons. 9 of those 30 do not have wings. So how many of our dragons are wingless? 9 divided by 30. Anyone know what 9 divided by 30 is? 0.3. So our Q squared equals 0.3. Hopefully you remember from the last example that if Q squared equals 0.3, we take the square root of Q and the square root of 0.3, and we're going to get Q equals 0.55. And hopefully you know that P plus Q equals 1. And if Q is 0.55, what is P? .45. Because 1 minus Q is going to equal P. 1 minus 0.55 equals 0.45. You could then calculate P squared. .45 times 0.45 equals 0.2. So those that have big W for wings represent 20% of our population. That 0.2. How many are heterozygotes then? Remember, to calculate heterozygotes, it's 2 times P times Q. So 2 times 0.45 times 0.55 equals 0.5. So 50% of our population are heterozygotes, 20% are big W, and 30% are little w, little w. And that's our dragon example for this slide. And we just did these genotype frequencies. Once again, this is giving you a second slide to practice with. You can use a chart like this to fill out and help you answer the questions. And hopefully you remember that your 2PQ stands for heterozygotes. So our heterozygous dragons are what 2PQ was. Now, if you're going to fill this out with your numbers, then you would know that your dragons that are heterozygotes are 50%. Here we have another dragon example. In this example, how many dragons did you find? Hopefully now that you've counted them, you've found 21 dragons. How many dragons do not have wings? There are 7 wingless dragons. Now, you have a little chart here to fill out as we go along. So we know we have 7 out of 21 that are wingless dragons. So 7 out of 21, 7 divided by 21 is 0.33. Where does that 0.33 go? If your answer was as q squared, you are correct, because q squared is our recessive trait. So when we have 2 of them, so that would be our ones without wings. So q squared is 0.33. What then is q? How do we calculate q? Hopefully you remember we're going to take the square root of the q squared number to calculate q. Now take your square root of 33, or 0.33, and you're going to find that it's about 0.58. So if q is 0.58, what is p? Well, p is, well, we know that p plus q equals 1, so p is going to equal 1 minus q. So that means it's 1 minus 0.58. So p is going to be 0.42. And when we square p, we do 0.42 squared. That's going to give us a p squared value of 0.18. Then what is 2PQ? 2 times 0.42 times 0.58 is 0.49. What percentage of your population are heterozygotes? 49%, because the 2PQ is 0.49. I've already told you that the Hardy-Weinberg equation does not have any real populations that meet its conditions, but our large populations we consider close enough for this to work. We also use it to determine when a population is not in equilibrium. We use this a lot in conservation biology work, in wildlife management. We use this to figure out how to properly breed animals together when we're working on an endangered species plan. Because we know that the genetic variation in a species will impact its ability to survive into the future. Having more genetic variation is always good. We also sometimes want to try and bring a species back into equilibrium. And that's one of the reasons why we utilize this equation, to figure out where things are and to figure out what we need to do in order to fix them. And populations are under selective pressures in nature. We see this all the time. In the top example here, in the top picture of mice, we have one dark mouse and a whole bunch of white mice. But over a lot of different generations, we see kind of the opposite in the population. A lot of dark brown mice, a couple white mice left. What do you think might have caused this shift? Anybody thinking that maybe they live in an area now where there's more dirt to be able to blend into? Or a place with more shadows as opposed to more sunlight? Because in a lot of sunlight, the white is good. But if you're in an area with a lot of shadows, then the brown fur is good. Or maybe in an area where your your predators don't have good eyesight for picking up dark colors. It's perfect to be a dark colored mouse then. So lots of generations can cause a shift in your P plus Q equaling 1, your allele frequencies, and your genotype frequencies. One in humans that we've seen change is lactase persistence, also known as lactose tolerance. In humans, the ability to tolerate and for your gut to digest lactose has increased significantly during the past 150 generations. But we find that it has not increased the same in every different people group. People of European ancestry tend to be the most tolerant to lactase, whereas people of Native American and Asian descent tend to be less tolerant of it. So it has something to do with the diets of our ancestors in the areas in which they lived, and which of them were able to successfully pass on those genes to the next generation. There are some examples for you to review. We will be going over these in class together, but I do want to give you the chance to work on these ahead of time. So we're going to quickly click through them together. So this is back to those 4 o'clock flowers. And in this one, you are considering a population of wildflowers where it's incompletely dominant. So a red and a white are dominant, but the pink is an incomplete dominant situation. You find one that has 320 red flowers, 160 pink, and 20 white. You're then asked to calculate the number of copies of each allele. Now you could do it the way that we had been doing it, where it was, okay, well, for the white, there's 20 of them, so we have 20 divided by the population, or if that doesn't work for you, you can calculate the copies this way. Then be sure when you do this that you're going to divide by the total number in your population, like you see here. So 80% are red, 20% must then be white. So the Hardy-Weinberg equilibrium describes a constant frequency of alleles in a gene pool. Consider an example where you have 500 wildflowers. If you have 500 wildflowers, you have 1,000 alleles. And you know that P is 0.8 and Q is 0.2, which is what we're seeing here. Your frequencies are 80% red, 20% white. So then you want to calculate, well, then what percent will be red, what percent will be pink, and what percent will be white. You do this by using the P squared, 2PQ, and Q squared. And for this, you already have the answers for P and Q. You just fill them in. 64% red, 32% pink, 4% white. Now you can double check this with a Punnett square if you'd like, but I'm not going to make you do this on the exam. This is once again that Punnett square from the last slide, and this is the math that goes along with it. Our final question is question 3. So we can assume that PKU is in Hardy-Weinberg equilibrium if our PKU gene mutation rate is low, which it is, and mate selection is random with respect to whether or not somebody carries PKU, which we assume it is. And we also assume that natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. For those of you unfamiliar with PKU, it is a genetic disease, but it is only triggered if they have exposure to the phenylalanine. If you can keep that out of their diet, then they're fine. We assume a population is large. Human population is very large. And we assume that migration has no effect and that all other populations of humans have similar levels of PKU frequencies. Okay, we can assume all of this. The occurrence of PKU is 1 per about 10,000 births. So that means Q squared is 0.0001 because Q is 0.01. So the frequency of P is 0.99. We can then figure out the frequency of carriers, meaning heterozygotes, for PKU. Less than 2% of the US population by this equation comes out to be a carrier of PKU. In class on Monday, we will be doing some more examples of Hardy-Weinberg and going back through some of these examples that we've done here. And I hope you have a great break.